Hello, everyone. Last lecture. Last lecture. Amino acids and proteins. Perhaps the most important biomolecule. The linear sequence of amino acids in a protein and the chemical nature of the amino acid side chains enable protein to perform their functions. So I read this statement because the linear, this one right here, it says the linear sequence and the side chain are what determine the function of the proteins. I mean, it really depends on the subsequent amino acid after the first one, after the second one, after the third one. And of course, what makes the next amino acid different is the side chain, or we're going to write here, as we learned in organic chemistry, the R group. The R group. So you've known about, you've taken classes that talk about proteins. What do? They catalyze reactions. We call those enzymes. They involve in chemical signaling, that we call those hormones. They are involved in storage, like myoglobin stores oxygen, structural collagen in skin and tendon. You hear about collagen commercials for natural or restoring natural skin, and then protective for antibodies, especially with what's happening now. Contractile myosin in the muscle for muscle movement, contraction, and relaxation, and then transport oxygen and hemoglobin. No wonder why it's the most important one. So we're going to begin and go ahead and move through this. And again, you can just rewind, go back and forth, and email me if you have any questions. So let's begin. Amino acids. What are amino acids? So the word amino acids means you have an amine. So that's N, H2, or obviously you can have NH3 if it's protonated, and you have a carboxylic acid. And of course, that's this. Now, the importance here is the amino acid is an organic compound that contains a quaternary ammonium group, that's this one right here, and the carboxylate group right here. So when it's deprotonated, the carboxylic acid, that's called carboxylate. When it's protonated, the amino acid is protonated, that's called a quaternary ammonium group. So you know that's ammonium. And here's your R group, that's the side chain. That's how all amino acids are different. Very well. For amino acids, the R group is called the side chain or the variant group. The side chain can be a hydrogen, hydrocarbon. You're going to see them in a second. I'm sure you're familiar with at least some of them. And they are named based on the identity of the R group. The R group is what makes or breaks things. So, for example, if you have the simplest, which is glycine right here, this is the simplest amino acid. And by the way, we usually say AA for amino acids. Okay, I don't know why this keeps, okay. Wow. For amino acids. Here it is. It's only an H right here. That's your R group. I know an R group is not exactly like an alkyl group, but we call it an R group in the, I guess, the biochemistry realm. And then you can have a CH3, that's a methyl right here. And then this would be called alanine. So once again, notice the difference between these two. There are 23 amino acids that make up proteins in plants and animals. 20 of them are directly specified by the genetic code in DNA. So we're going to worry about those 20 amino acids. And those are called the common amino acids. Those are the 20. All 20 are alpha amino acids. This is what alpha means. Okay. They have, again, you have a side chain to it. It's a carbon attached to the ammonium, and the carboxylate, and the R group. Obviously, there's an H on it. And then they are called alpha amino acids because their side chains are attached to alpha carbons. So this is your alpha. If you recall what alpha means from before, you know, when we did the enolates, all of that stuff we did before. Here's your alpha. And then you have a beta afterwards, so on and so forth. So remember, that's your alpha carbon. That's your alpha. So recall what we did. So that's the definition. Here are the 20 amino acids. Now, obviously, they're there in front of you, but you should know them. It's helpful to know all of them. So you start with glycine, and it's critical that you know the abbreviation and the structure. So if you look here, you have glycine has an H, alanine has a CH3, and notice that it has a 3-letter abbreviation, like GLY for glycine, ALA for alanine, valine is if you have we should know what this is. This is a isopropyl group. Then you have, of course, more alkyl groups like leucine, isoleucine. Iso, remember how that's related to it. You have proline if you have a ring. You have glutamine. You have cysteine if you have a CH2, CH, so on and so forth. All right, and notice the abbreviation. Now, before I move on, you should be able, if you ever see, you should be able to transform this into a skeletal structure. There's no reason why, you know, all of a sudden you say, I've never seen this. So here it is, glycine, we have an NH3. Here's our alpha carbon. What's attached to our alpha? There's an H, so we don't have to show it. And then we have a carboxylate. Okay, that's a minus, and here's our double bond. So here's our alpha carbon right here. That's our alpha carbon. Here's the other one, the one down here. Minus there, and of course we have a CH3. That's this one right there. That's our methyl. So on and so forth. So again, be able to really, you know, go back and forth between these. That is expected of you. So we learned this in first day in organic chemistry. Charges on amino acids. The charges on amino acids are dependent on the ammonium group or the amine and the carboxylic acid or the carboxylate. So the structural formulas of the common amino acids all contain at least one carboxylate group and one quaternary ammonium group. Okay, so what do you have here? Here you have a plus and you have a minus. So the overall is what? It's a 0. Okay, they cancel out. So overall charge is 0. So, but we have to take into account the pKa as it's related to the acidity and basicity, remember, of compounds. pKa's are very critical. We know that if we have a carboxylic acid and it's in the acid form, then in the acid form between 2 and 5, that's acidic, that H will be attached, so it's in the acidic form. It engages in an acid-base reaction, loses that H to water, and now it turns into the basic form. So what you need to be aware of is this range right here, the pK of 2 to 5. It depends also on the R group. From the Henderson-Hasselbalch relation, there's an equation you learned in Gen Chem 2. We know, and we're going to examine it shortly, we know that when the pH is less than the pK of a carboxyl group, then the carboxylic acid form, this one right here, is predominant. And when the pH is greater than the pKa, then the carboxylate ion is predominant. Okay, so this is critical right here. So if you have for the carboxylic acid, if the pH is less than the pKa, okay, I guess I'm going to put it on this side here too. Okay, so this side right here is if the pH is less than the pKa, and remember the pKa has to be between 2 and 5. Then that's what you have. If the pH is greater than the pKa, 2 to 5, then that's what exists, this one right here, this form. Likewise, the ammonium group is in equilibrium with its conjugate amine group. We know that the pKa is about 9.5. And when it loses a proton, so it loses, let's say, this proton right here, it's gone, notice, an acid-base reaction becomes an amine. So now the following is the result. Okay, so this is the key, the pKa right here. So if the pH of the solution, right here, pH of the solution, okay, is less than the pKa, which is 2 to 5, sorry, not 2 to 5, 9.5. Just like we did above here, then this form exists. But on the other side is if it's larger, then it's going to be, keep saying this, 9.5. Then this form exists. So what is key? What do you need to know here? You need to remember two things. You need to remember those pKa's, 2.2 to 5 for the carboxylic group. And you should be a pro at this. You know that the pK of a carboxylic acids are between 2 and 5. pKa's of the ammonium is about 9.5. Just use, and then the second thing, what do you need to do? Well, just use really your basic chemical knowledge. A low pH means it's acidic. So if it's less than, okay, if it's less than the pKa, then it makes sense that it's still acidic. The lower the pH, the more acidic it is. You know that, right? From 1 to 14, the scale. That anything below 7 is what? Acidic. Anything above 7 is basic. However, we have two ranges here. So if it is low, so for example, if my pH was a 1.3, then it makes sense that it's more acidic, so it's going to stay in this form. Or if it's actually close, right, or equal to, let's say. Same thing here. Well, what if I have an 8, a pH of 8? Then it makes sense that I have, again, this form, the basic form. Okay. So the amino acids, since amino acids involve the carboxylate group, conjugate pair, these, and the quaternary ammonium group, amine group conjugate base pair, these, then the total charge of the dominant form of the amino acid will depend on the pH. So we're going to do very, very basic, seriously, very, very basic exercises here. So for example, look at this. You have alanine. So this is its physiological in the body. This is its physiological pH. It's about 7.4. So if we at physiological, that's the physiological pH, you know, the blood, it's about 7.4. The pKa of the amino acid carboxyl group are between 2 and 5, depending on which amino acids. And therefore, 7.4 means it's in the basic form. So here it is. This is what I have. Why? Because again, the pKa should be around 2 to 5. And if I have my pH at 7.4, it means it's greater than the 2 to 5, which is the pKa. So it makes sense that it's going to be in the basic form. Go back up and look at it again. Here it is. Now, if you have a pH of 7.4, because you can't have multiple pHs, it's in the solution, and that pH is actually less than the pKa of the ammonium group, which is what, 9.5. So then it stays in this acidic form. Look over here. The pH is less than the pKa. Now we're just going to, again, go with basic things. If I were to make everything, all of the side chain really involved, then it would become really more complicated. But we're not at that point. You'll learn that in a more in a biochem class based on your knowledge of chemistry. So the predominant form then has a negative and a positive formal charge on the ammonium group, which gives it a total of 0, as I said before. This is what we call a Zwitter ion. Zwitter ion meaning it has an amino acid with a charge equal to 0. And the reason why is because you have two opposite charges. You have two opposite charges. You have the negative and you have the positive. So you have +1, -1, you get a 0. So that's what Zwitter ion means. Okay, and it's a hybrid. You can think of it as, you know, you have opposite charges in there. Okay, the amino acid structure in the table provided earlier are the predominant forms at physiological pH. Insufficiently acidic, so the predominant form again are physiological pH. So the ones that we showed you on the table. In sufficiently acidic or basic solutions, the total charge of the predominant form of an amino acid will change from its physiological physiological value. So depending on the concentration of the solution, whether it's acidic or basic, if it is acidic, then the total charge will shift. And we'll show you this in the next example. So we're not talking about physiological pH anymore. We're talking about a pH of 1. And you know, pH of 1 is extremely acidic, very acidic. So therefore, the pH here is less than the pKa, which is 9.5. So therefore, it's going to stay in this form. That's 1. The pH is 1. 1 is less than 9.5. And 1 is also less than 2 to the 5. pKa. So that is also going to stay in the acidic form. So you actually have now what? Both in their acidic forms, simply because, again, it's extremely acidic. So what is the overall charge then? Okay, well, we look here. We have a neutral. There's no charge on this O, but we keep going and we see a positive. So therefore, the predominant form, okay, has an uncharged carboxyl group, which here is uncharged. And then it has a positive one on the ammonium or the amine. So therefore, the total charge is positive 1. Let's look at another one. What if we actually now shift to extremely basic? 12 is extremely basic. So then what's going to happen is 12 is larger than 9.5. So this will end up losing the proton. And the pH is also much larger than the pKa here, which is 12 is much larger than 2 to 5. So this also will end up losing its proton. And therefore, overall, it's going to have a negative charge. Why? Because this is neutral. And this has a -1 charge right here. And that gives it an overall of a negative. We're going to do a practice problem. We're going to draw the predominant form of valine when the pH is 7.4, also at 1 and at 12. And then while we're doing that, we can determine the charge. So valine. I suggest you going and copying it. So go and copy it, or I'm about to obviously draw it, but it has an isopropyl group right here. So we're going to draw it right here. Now I'm definitely going to need more space, but we'll do our best. Okay, so we're just going to do physiological. So we're going to do, okay. So this is valine right here. Now 7.4, 7.4, 7.4. So if the pH is 7.4, it means it's larger in this case than the pKa of the carboxyl group, COOH, which is, remember, 2 to 5. So therefore, so if you recall, go back up here. The physiological pH is 7.4. So the pKa of the carboxyl group was 2.2 to 5. So therefore, to draw this, we need to remove this H because it's more basic. So we're going to remove this H and we're going to put a negative charge. However, this 7.4 is less than 9.5. So that means it's going to stay in this basic form, an acidic form. So it's fine like this. What is the overall charge for D? Well, we have a negative, we have positive. I'm going to circle them so you can see them. Positive and negative, the overall charge is 0. That's the overall charge, or the total charge. Okay, so this is A and D. I know I'm jumping because D is related to A. Let's do B now. So you can begin by drawing this as physiological and then you can draw the skeletal structure. In fact, I should have drawn the skeletal structure, but it's okay. Okay. What pH do I have? I have a pH of 1. Well, 1 is less than 2 to 5 for this side. So that means it's acidic and it's going to stay in that form. Okay. 1 is also less than 9.5, which means that this can stay also in its form. So then the overall charge is a +1 because, again, this is neutral. So +1 is the total charge. And that would be B and E. Finally, F, sorry, not F, C and F. Is we have a pH of 12. Okay. So once again, draw it at physiological and then you can make the changes or you can just figure it out before you complete the structure. Okay, let's start. All right, so if we have a pH of 12, it means it's highly basic, very, very basic, which is larger than 2 to 5. That means this is going to lose its proton. So you'll have a minus. And then this will also lose its proton because it's also above 9.5. So 12 is larger than 9.5. And so the overall charge will be -1. Good. So amino acids are classified by the polarity of their side chains and the ability of the side chain right here, if you see, to acquire a charge. To acquire a charge at physiological pH. Look here. So the polarity plays a big part. Nonpolar amino acids with nonpolar side chains. Those are called hydrophobic side chains. The zero is the zero. It means it has a charge of 0 at physiological pH. You have polar neutral, also 0 at physiological. Polar acidic and then polar basic. All of these are polar, hydrophilic, but again, now when you get to the acidic and the basic, the charges can become negative or positive. So let's start. You should know what are the nonpolar amino acids. And they are all of these: glycine, alanine, valine, proline, phenylalanine, leucine, isoleucine, methionine, and tryptophan. All of these are nonpolar. And the reason why they're nonpolar is you have what? All you have is hydrocarbon chains, okay? Carbon, carbon, hydrogen. Okay, we know that all of these, anybody from basic chemistry class know now that all of these are nonpolar. Okay, look. All of these are nonpolar. Once again, hydrocarbons. So nonpolar, as they say, side chains. Polar neutral, you should know them: glutamine, asparagine, tyrosine, cysteine, serine, and threonine. Okay. Threonine, all of these are polar but neutral. So polar but neutral. There's no charge, uncharged. And the reason why they're polar is because, you know, you have a carbonyl, you have a carbon-nitrogen bond. All of those are polar bonds. You also have an OH, 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 SH, could be polar. Or it is polar. Polar acidic amino acids. Okay, this now changes. So what happens is amino acids have a polar hydrophilic side chains, and their predominant forms have side chains with a negative formal charge at physiological pH. So at physiological pH, right here, because it's a carboxylic acid, it becomes negative. So you have aspartic acid and you have glutamic acid. So all of these are, again, acidic amino acids. And they have a negative charge. Notice the negative. It's a negative charge. There's a -1. Here it is. -1. There it is. So it says polar acidic amino acids are given the acidic term in their classification because their acid forms are stronger acids than those of the polar basic amino acids. So they will be discussed next. So again, they are given this because their acidic form right here is stronger than the polar basic one. So if you see here, you have the polar basic amino acids. You have arginine, lysine, histidine. And they have a polar hydrophilic side chains. And except for histidine, their predominant forms have a +1. Here it is. N with 4 bonds is positive. N with 4 bonds is positive. And where does the formal charge come from? Is the quaternary ammonium group. There you go. The polar basic are the following. So like we said, arginine. All right, so let's look here. Properties of amino acids. Although some amino acids contain hydrophobic side chains, overall they are water soluble. So they're water soluble. All amino acids are hydrophilic due to both the presence of polar covalent bonds that are capable of forming polar hydrogen bonds with water, and the fact that they carry charges. So right here, you can make hydrogen bonds, OH. Also this. These are H bonds. They form H bonds with water. Plus, they can carry charges, and anything that's charged is soluble in aqueous solutions or in water. So all of them are water soluble. Stereoisomerism of amino acids. So amino acids are chiral compounds, and the reason why is because this R group, the alpha, has the 4 different groups. Here they are. 1, the H, the R is a different group, the carboxylate, and the ammonium group. All of those are 4 different groups. So that's why they are chiral. And if you remember what chiral means, again, they have enantiomers. So if you look here, you have L amino acids and you have D amino acids. So if you have the L amino acids, that means the NH3 is on the left. So the best structure for this is to actually use the Fisher projections. Okay, not sure what this is. Okay, if you have NH3 on the right, NH3 on the left, and that again affects the name of it. So in this case, you have the NH3 right here is on the right. So that's an L amino acid, D amino acid, I'm sorry. If you switch them, where it's now the NH3 is on the other side, then that would be an L amino acid. So what is the feature projection of alanine? Well, you have to remember the R group. The R group is a CH3. What are we looking for? The D and the L. So the D means NH3 is on the right. So this is your D, alanine. This, and your L-alanine is the opposite. Right here. Okay, we're reaching now the most important portion, which is peptides and proteins. The peptide bond. This is the most important thing. The peptide bond, by the way, is nothing but an amide bond. So organic chemists like to call it an amide bond because that's what it is. But in biology and biochemistry, they call it a peptide bond. Well, what is it? Well, you should know what an amide is, number one. That's going to help you. Amide is this. You know, you could have had NH2 or NR2, whatever it is. But this is the bond right here. That's called an amide bond because this whole thing is called amide. We did that in the functional groups. So peptides and proteins consist of amino acid residues joined by peptide or amide bonds. Here it is, self-explanatory. What is happening? The step one is you have one amino acid and you have another amino acid. What happens is a reaction takes place between the charged oxygen on carboxylate and the two H's on the ammonium. You know that what's going to come out, you have an O and two H's. That's water right here. So then you add them together, you put them together. Reaction is going to take place. And when you put them together, you form a peptide bond right here. A new bond is made between the carbonyl, carbon, and the nitrogen. And here it is. Here it is. So I'm going to show you the peptide bond. There it is. That's the peptide bond. The peptide formed in this example is called a dipeptide because it contains two amino acids. Here's one and there's another one. So that's how a peptide bond forms. There it is. It's better than me. It highlighted it for you. So let's draw a dipeptide between valine labeled a peptide bond. Okay. Start with the NH3 on the first valine. Here's my alpha carbon. I have an H on it, and then I have a CH, CH3, and a CH3. Here's the first one. This is now the second one. Okay, here we go. Here's the first valine, here's the second valine. The reaction is going to take place. So remember, we're at physiological pH, so we need to put the minus on these. Okay. The reaction is going to take place between this and these two H's. Okay. How's it going to look like? Well, let's do it. So I'm going to redraw this. I'm going to make sure you put all the charges, because even I here, I'm forgetting them, but not on purpose. So make sure you remember all the charges at physiological pH. Okay. This is gone. So now the bond is going to form between this C, carbonyl, and the nitrogen. Oops, sorry about that. Forgot about them. Put this one down. Okay, where's my peptide bond? Here it is. So I have a dipeptide now. Good. Well, if you have formation of larger peptides, we're going to end up drawing this. The formation of a larger peptide is by adding more amino acids one by one to a growing peptide. That's how proteins grow. They are, again, multiple amino acids bonded together via the peptide bond. So you begin with a general form of the dipeptide and then add a new amino acid residue. The new peptide can be made using the same two steps as we did before. Nothing different. So here you had a dipeptide. You want to make it a tripeptide. You add another amino acid residue. So you add it just the same way, and now you have a tripeptide. Here's the first one. Here's the second one. And here's the third one, including that N. So third one. First one, and then the one I didn't circle is the one in the middle. Peptide terminology. You have the end of a peptide formula that has a quaternary ammonium group is called the N terminus, and the end that has a carboxylate group is called the C terminus. So here's the N terminus. The first amino acid starts with the ammonium group. That's called the N terminus, terminal at the end. And then the C terminus is right here. And the peptide pattern around the bonding pattern is called a peptide group. And this is really key. I can't really do anything other than this. I think this is very, very clear. So if you're asked, well, what is a peptide group? You are to circle the carbonyl plus the amine. Here's the carbonyl and here's the amine. That's what you include. That's called a peptide group. That's a peptide group. Note that the nitrogen, the peptide group, does not have a +1 formal charge, as does the nitrogen at the N terminus, right? So the formal charge makes sense. This N, the bonding pattern of it allows it to be neutral. This one doesn't. So you have to make the distinction between them. Peptides are identified by the use or of a common name or by listing the amino acid residues, 3-letter abbreviations in the order from the N terminus to the C terminus. So for example, here I have a pentapeptide. A pentapeptide. Penta means what? 5. And it's given to you in valine, right? And then what do you have? Ferritic acid, alanine, arginine, and glycine. Once again, it is key that you remember these abbreviations. This pentapeptide is drawn by forming peptide bonds between the predominant form at physiological pH, therefore resulting in the peptide at the physiological pH. So you need to be aware that the charges here now influence the whole thing. So you have a +1, you have a -1 there. So you have to take these into account when you do it. Be aware of this. So let's try this one. We need to draw the structure of the predominant form at physiological pH. You should be able, by the way, to draw it at any pH I give you. So we're just going to do it on physiological pH, and then you can make the adjustments. So we have glycine. And again, I'm going to write as small as I can because this is going to take a large space. Okay, now here's my glycine. It's attached to now, it starts with the N terminus. Now, this is my glycine. Here it is, GLY. Okay. Now, what's next? Lysine. Okay, so where is lysine? Where is lysine? Where is lysine? Here it is. So sometimes I put these as, you know, the skeletal structures, but we can just show this, but again, it's going to be a large. So you have 1, 2, 3, 4, right? Carbon atoms attached to an ammonium group. Okay. So this is my lysine. What's next? Tyrosine. Let's look at tyrosine. Here it is. I have a benzene ring in it. Okay. And so this is my tyrosine right here. And then our tyrosine. And then finally we have alanine. I'm not sure if you remember, we just mentioned this. This is only a CH3. Okay. Well, here it is. What is the overall charge? Well, let's go. We have a plus there. We have a plus here. And we have a minus there. So we have a minus there. So what's plus 1? 1 plus 1 is 2. Minus 1 is plus 1. So the overall should be a plus 1 charge. Again, you have to look at the charges on the entire molecule. Okay. So we're getting close. I'm just going to go, really, a lot of this is really just more factual, but you still need to know it because it's going to prepare you. Just quickly, an example of biologically relevant peptides. A protein consists of one or more large peptides and has a specific biological functions. So that's the key, right? You can have a peptide or you can have a large peptide, like you can have, not sure, 100, etc. But if it doesn't have a function, biological function, then it's not considered a protein. So although shorter peptide chains, less than 50 amino acids, have specific biological functions, they are generally not classified as proteins. So those short peptide chains function as chemical signaling compounds. Over 100 of them have been identified. So again, they have to be large peptides, very large peptides, and they have to have a specific biological function. But again, that's why these go together. They really go together. They have to be large peptides, and they have to have a special or a specific biological function. So they can't be short peptides and the biological function. That's still a peptide. Or they can't be large and there's no biological function. Has to be one. Endorphins are examples of chemical signal peptides. They're natural painkillers. So when you exercise, they actually improve the mood, and they interact with receptors in the brain to inhibit the transmission of pain signals. 5 endorphins have been found, and one of them is alpha endorphin. It contains 16 amino acids from the N to the C terminus as shown, tyrosine, glycine, etc. Like this is here, and alpha, one of the alpha endorphins. Another example is oxytocin. You know what that is. It's produced by the pituitary gland, stimulates the uterine contraction and labor, and it has about 9 amino acid residue right here. And it was the first naturally occurring hormone to be produced in the lab. Very well. So now what do we get? Where are we getting? We're getting to also another important aspect, and it's the protein structure. I believe you're familiar with pretty much a lot of these, but I'm going to elucidate as much as I can. So we're going to go quite fast through this. Primary protein structure is simply the order. It's a sequence of those amino acids. So it's like valine, asparagine, alanine, arginine, glycine, aspartic acid, whatever it is, whatever comes in it. It's just the chains. It's just those chains of those peptide, the peptide. That's all it is. That's the primary structure. So that's the foundation, is you have to have amino acids to create a protein. So you start with your amino acids, and they have to be in that order too. So that's called the primary structure. The primary structure in protein is analogous to arrangement of letters in a word. So for example, just because, you say, well, how about I switch asparagine with valine? Still the same thing. No, it's not. It is not the same peptide anymore or protein. It has to be in that order. So for example, these words right here, They have the same letters, but they're not the same words. Edit, not diet, is not tied, is not tied. So once again, it has to be in that order. Different proteins typically contain about 40 to over 400,000 amino acids. There are 400 distinct dipeptides, 2 to the 20. There are 8,000 distinct tripeptides, 2 to the 30. 20 to the 3, I'm sorry. That's 20 times 20 times 20. That would be 8,000. And when there are 100 amino acids in the chains, there are, I mean, this is just the mind-boggling. If you have 100 amino acids in a chain, you can have 2 to the 100. 100, by the way, is called Google, by the way. But it's spelled this way, Google. Okay. You get 1.27 times 10 to the 10 to the 130. This is a number that you cannot imagine how large it is. This is much larger than Avogadro's number, which is 6 times 10 to the 23. So it's key that you have those arranged in that order. So let's do this understanding check. Write the names, and you should do this, of all the tripeptides that can be made by combining one glycine, one alanine, one aspartic acid. For example, you have glycine, alanine, aspartic acid. If you want to try it, you should pause now. Okay, so I'm going to continue. You pause. Let's check again. We did this. We can do now glycine, aspartic acid, alanine. We can do now the alanine, glycine, aspartic acid. We can do alanine, aspartic acid, glycine. We can do aspartic acid, glycine, alanine. We can do aspartic acid, alanine, and glycine. Oh, what happened here? Okay, I don't know what happened. Let's see. Uh-oh, this made us jump. Okay, so far, how many do we have? All right, so we have this, that. Okay, can you get any more? So we had glycine, alanine, aspartic acid, glycine, aspartic acid, alanine, alanine, glycine, aspartic acid, alanine, aspartic acid, glycine, alanine, so on and so forth. I don't think you can continue. So you have 6, right? Try it if you are not convinced. So you have 6 different peptides, tripeptides. Secondary protein structure. The secondary protein structure depends not only on the sequence, just like we said, but also on how they are folded. Secondary protein structures, they describe the geometric patterns that occur when individual peptide chains fold back on themselves. So we're talking about this. So basically what they do is they go now in a helix. They go in a helical structure. And secondary structure results from hydrogen bonding between the peptide bonds within an individual peptide. Okay, so let's elucidate. Let's start with the alpha helix. So you're starting with the N terminus right here. Here's your first amino acid. Where you see this yellow on that carbon, that's where the amino acid begins. So be aware of that. So if you see here, you have a first amino acid, second amino acid, then you have the third amino acid because it's twisting, fourth amino acid right here, fifth amino acid, so on and so forth. The way it's twisting right here, that's called an alpha helix. And you have hydrogen bonds between them. You have hydrogen bonds occurring between the carbonyl carbon and the hydrogen on the nitrogen in the in that amino acid. So it resembles a spring. So if I give you one like this on a test, you should be able to really put those hydrogen bonds, mark them. The beta sheet is another form. And the beta sheet occurs when a peptide folds back on itself in a side-by-side arrangement. So basically, you start with the N terminus right there. Keep going, keep going. Keep going, keep going. So that's called now a beta sheet. This is a beta sheet right here. Once again, there are hydrogen bonds between them. There are hydrogen bonds between them. So side-by-side arrangement, side-by-side arrangement. In addition to alpha helices, there are also a few other, much less frequently, and this is key. They categorize as secondary structure. They're rare, but we're not discussing them. You got beta bleeded sheets, etc. A key feature of the secondary protein is that it only involves hydrogen bonding between peptide groups within an individual peptide chain. So again, this is very important to remember. Tertiary structure. Now, number one, we have to get into, you'll see this a lot, especially with the situation we're in, and they show a protein structure, and you'll see these helices and these twists, and you'll see like these sheets. Well, now you should know what these are. These are your alpha helices and your beta sheets. So what is now the third, the tertiary structure? The tertiary structure is of alpha helices and or beta sheets. They are basically, what they do is they fold now, and you have a collection of them. It can't just go on and on and on, or keep like in a helix, you know, like a spring that is like very, very long. It keeps extending. What happens is these proteins, okay, they fold onto each other. Okay, it's kind of like, you know, when you if you make it's like a, if you can't extend something, right, and keep extending it indefinitely, it has to eventually fold on itself. And if you see here, all of them are amino acids and alpha helices or beta sheets, but they are folding. Okay, they fold into a more compact shape. Again, nature is smart. Nature also wants to occupy a small space or keep, you know, stay efficient. The compact shape of the peptide is called a tertiary structure. So when you have that compact shape of the tertiary structure, that's called of the protein, of the peptide, that's called a tertiary structure. And we use ribbon models. Those are called ribbon models. So you have the ribbon-like to represent the geometry of the secondary structures. So here's your secondary structure of alpha, beta sheets. And then you have the flat side by side. Notice how they are side by side, beta sheets. And sometimes arrows are used at the ends of the ribbon to indicate the direction. So if you see this arrow right here, or it's going there, you see that, it says to you that you're going from the N to the C. You're starting from the N terminus to the C terminus. So for example, here's the ribonuclease A. RNase, it's an enzyme used to break down RNA, is shown on the right here. Well, how does this protein structure how does it interact with each other in a more compact form? Well, this is all intermolecular forces. Okay, so intermolecular forces. When we teach this in chemistry, I always tell students you will see how important this is, and here it is. Well, of the many folding patterns, conformations possible for protein, there's usually only one that leads to a native biologically active molecule. So there's one conformation or pattern that leads to this biological or this protein having a function, a biological function, biologically active. The sequence of the amino acids usually determines the folding pattern. So the primary structure is key here. So both secondary and tertiary depend on the primary structure. Some of the interactions that are involved are tertiary structures as illustrated below. Here it is. For example, let's say it begins from here. Beta sheets. Well, what do we have here? We have hydrogen bonds. You continue, have an alpha helix. Okay, well, we have to pay attention to the other ones, and you have to know all of these interactions. You have hydrophobic interactions. So all of these are when you have the non-polar amino acids. These are London dispersion. We just call them hydrophobic interactions, London dispersion forces. Right here. You have hydrogen-hydrogen bonds. We know what they are. You also have hydrogen-hydrogen bonds. Now you have a salt bridge right here, a salt bridge. Salt bridge is, again, you have a negative and you have a positive attracted to each other. You know that. And you can think of it as an ionic, okay, and an ionic bond. Okay, so you can think of it as an ionic bond. Negative is attracted to the positive. You have also hydrogen bonds and hydrogen bonds. You also have one more here, very key. It's called the disulfide bridge. Okay, so when you have cysteines, two cysteine amino acids, those side chains are called, are made up of sulfurs and attached to CH2. What they do is they form a bond between them. That's called a disulfide bridge. Okay. Here it is. Here are the explanations. We talked about them. London dispersion. They're water-free pockets. Hydrogen bonding. We know what that is. Salt bridges. Those are non-covalent as one of the covalent. Don't worry about chapter 4. That was, again, maybe if you didn't go last semester, but just remember those are non-covalent interactions, meaning, you can think of it as the ionic. We usually, just an easy way to remember it, but we usually say that a salt bridge is between the formal charge on a polar basic and the negative on the polar acidic. That's what we mean by the salt bridge. But they're like ionic bonds in a sense. Disulfide bridge is if you have cysteine, the thiol groups, and dipole-dipole and ion-dipole forces are also common. So you have to know all of these interactions. Now, I'm going to just talk quickly about I know I'm going a little bit longer here, but the rest of it is really for you to really read and still know it. Globular proteins, hemoglobin, you know what those are, albumin, fibrous proteins, all of these you have to know the distinctions. But I'm just going to elucidate the final one because sometimes students have a hard time understanding it. And I have to admit, when I was a student, I struggled not knowing the difference between the tertiary and the quaternary structure. A large number of native proteins are a combination of more than one polypeptide chains. So for example, hemoglobin. Hemoglobin is more than one polypeptide chain. Quaternary protein structure is the overall shape that occurs when two or more individual peptide chains assemble to make a protein. So you can think of the tertiary as like one peptide chain. It keeps going and going and going, and it's an interaction between them. But in a quaternary, the fourth dimension you can think of it is that it's actually multiple peptide chains. So when they're composed of two or more, those are called subunits. So you'll have one peptide that's called a subunit, another peptide is called another subunit. So the structure, is usually depicted here, space-filling models. So like you can see, like this is one subunit, there's another subunit, and the interaction between them. The forces that hold the subunits together in the quaternary structures are the same as one involved in tertiary structure. I guess the best way, and all of these are pictures, real, like, space-filling models of proteins. Is to really look at this picture, which I found online. Very, very helpful. Primary is just the chain, the sequence. Secondary is the beta pleated sheets, the sheets, and the alpha helix. A tertiary is, once again, the three-dimensional folding. It folds on itself due to the interactions, chain interaction. A quaternary it's a protein consisting of one or more amino acid chains. So if you see, like, here's one chain and there's another chain, and then when they interact with each other, then that would be the fourth protein structure, or the quaternary protein structure. So know the difference between them and read the rest of this. These are good examples, like globular proteins. And then you have myoglobin, to carry oxygen, store oxygen, like in hemoglobin, albumin, and plasma. And then you have fibrous proteins on the other side. Those are string-likes, and you need them obviously for example, hair, collagen fiber, and keratin for the hair. So that is all, just to give you an idea, and that's it. Okay, thank you for listening. Ladies and gentlemen, that's it. That's the end of the course for the semester.